Hello and welcome to a very special series of Dive into Deeptech MoSS, brought to you by Republic of Work and powered by Helixworks Technologies. I'm your host, Aishna Gogia. For those unfamiliar with MoSS, it stands for Molecular Storage System. In other words, using DNA. Yes, you heard it right. DNA as a data storage medium. We're excited to explore the incredible possibilities of storing data on DNA for various purposes beyond digital archival. Throughout this series, we will be talking to experts and innovators in the field to get their insights and perspectives on the potential of using DNA data storage. We will introduce the basics of DNA data storage, discuss various technologies used to store data on DNA, address the challenges in using DNA as a form of data storage, and explore its potential applications in different industries. So join us as we dive into the exciting world of DNA data storage and explore the potential of this revolutionary technology. I'm so excited to bring another episode of this series. Today, we're joined by Dr. Thomas Heinis, a professor in data management at Imperial College London, where he also leads the Scale Lab as well as the research in DNA data storage. In today's episode, we would be diving into one of his key projects, OligoArchive, the trends and opportunities in this field of technology, and discuss the impacts that could come from research labs working on DNA data storage. So let's get started and find out what exciting initiatives and project Thomas is leading ahead in this field of research. Thank you so much for joining us, Thomas.
Oh, my pleasure. Thank you. Hello.
So, Thomas, you have a PhD in computer science and you've been driving the research on developing tools to process big scientific data. Can you tell us a bit more about what made you get into this field of research and how did you transition into DNA data storage and data processing?
Oh, I always like to, or love to even work on kind of between disciplines. I worked a lot with neuroscientists, astronomers, biologists, etc., to kind of manage their data. And, a couple of years ago, I learned about DNA storage, kind of the promise or the idea of storing arbitrary data in synthetic DNA. And I thought like how exciting that could be. I kind of came up with a couple of ideas around it, what that could mean with my background in databases, on how we could bring these fields together. I came up with a couple of ideas and then we started trying them out. And that's where we are.
Perfect. And Scale Up is working in various projects and initiatives related to data storage, management, and processing. What areas of DNA data storage is your lab most interested in?
When it comes to DNA storage, I think we focus mostly on the computational aspects, right? Kind of like how do we map data from binary formats or arbitrary formats into DNA, and how do we read this information back? We focus a little bit, like I said, on the computational aspects because we are also located at the Department of Computing, and most of us have a background in computer science. But we also try to venture a little bit more into the aspects of biological aspects, chemical aspects, optimize the processes involved.
So I do have a lot of questions around that area, but, let's get started and let's talk a bit more about, the project, the OligoArchive project. So in 2019, you received funding under the Future and Emerging Technologies for OligoArchive along with other consortium members. Could you briefly explain what OligoArchive stands for and what are some of the objectives within this project?
So the name derives basically from the idea that we use oligos, DNA oligos, to store information. And when we look at DNA as a storage medium, it's mostly suited for long-term archival of data. That's where the name then comes from oligo and archiving of data, right? And the goal of it is quite simple, right? We wanted to develop a kind of like integrated end-to-end solution of going from input data, whatever it is, images, databases, etc., translate this efficiently into a representation of DNA, synthesize that, store it for an amount of time, then sequence it to read it back and convert it back to binary. So we really wanted to have this ability to go end to end with writing of the DNA, reading of the DNA, and then also having the capability of processing the data that is stored in DNA directly.
So what kind of progress has been made so far? Like what are some of the results from the partners within the project?
So, there's so many results. Where to begin? So essentially what we have done so far is we developed encodings. How do we efficiently encode data, imaging data, for example? One partner, Université de Nice, has worked on how do we efficiently encode imaging information in DNA and how do you read it back efficiently. We have worked on how can we encode information or relational databases in DNA, and how can we read that back as well, and also how can we perform certain operations of the data when it is already stored in DNA. HelixWorks has worked on a efficient synthesis technology for it and has developed a prototype of that. And we also worked on the sequencing aspect. How can we accelerate sequencing such that we can read the information back more efficiently?
That sounds really interesting, and even on the collaboration bit as well. So we'll probably dive into that now. So talking about the architecture for OligoArchive, so database engines is basically a software that enables database management system to create, read, or update and delete data. So they use a 3 storage hierarchy. What is database storage hierarchy, and how are you integrating DNA in the data storage, in the database storage hierarchy? And how is the OligoArchive architecture altering the DBMS storage hierarchy?
So what we try to do, right, whenever we develop some system for a database system, right, we always have to look at how do we actually store the data on a physical level, right? How do we store, what's the medium at the bottom? How fast can we access it? How precise can we access specific information in this medium, and so on. So that kind of drives how we develop a database management system. And here we thought, rather than storing information on a disk as one normally would, we're going to replace that disk with DNA. What does that then mean? What can we improve about the database management system? So we basically took a database management system as it exists, as it is available, or as open source. We ripped out the assumptions that this is stored on disk and plugged in DNA, and then kind of try to adapt the software such that we can actually store data in there efficiently and also perform queries on top of it.
And how does it stand against the widely used database storage hierarchy?
So the difference isn't huge, except that, is in the sense that the medium disk and DNA are rather similar, except that DNA is much slower to access. And it's also, to write and to read.
Okay. So ideally, the existing engines, the database engines, they have well-established procedures and tools for archiving data. And one of the objectives of OligoArchive, like you mentioned, is to leverage the database knowledge in optimizing DNA encoding and decoding. And I know I'm getting into that area where it's really hard to explain without going in depth. I have a series of questions. They're all interrelated, and you can probably answer them all in once or take it one by one. So the current database engines have well-established procedures and tools for archiving data. Is there a different database engine for OligoArchive? Because I remember reading an article, it was by ZDNet, that had all in bold that forget silicon, SQL on DNA is the next frontier for databases. Could you tell us about, the archiving and recovering tools for this architecture and the extensions required for implementing OligoArchive architecture?
Yeah, so what we did in the first attempt, right, we really tried to kind of replicate what the database does on the DNA, right? So what we did is we took Postgres, open source database system, and adapted it for DNA. And one thing that we exploited is that, in a relational database, when you have tables, etc., that have a certain structure. And we can exploit that structure. The common tools as they exist now for Postgres, they don't really do that. They just take the table, store it as a binary file, and then read it back. Because the underlying storage media doesn't care on how it's really stored. But when we take DNA, however, if we store in DNA, we can exploit the structure that we have in the data and compress the data to some degree. So, for example, when we store a lot of data in DNA, we have to store it in thousands, if not millions, of small, short oligos, right? So, if we take a file of a megabyte, this is going to go into millions of these oligos, right? Or has to be stored on millions of these oligos. When we then read the information back, we need to kind of assemble this file back together, and that requires information about which part of the file is stored in which oligo. Right. Now, when it comes to relational databases, the tables have a structure. They have certain fields. They also have what is called a primary key that helps to kind of tie the table together. And with this information, we can exploit when you store the information of DNA to reassemble the table in the end. If that makes sense, right?
Oh, yeah, 100%.
And we can also exploit there, right? When you use a database, there's a process called data cleaning. Okay. And basically assuming that we have certain errors in the data, we have a bit of dirty data, and that's exactly how the data comes back when we read it from DNA, because there's some slight errors there that we can compensate for through error correction, but still there's going to be a few errors in there. And this data cleaning process, for example, we can exploit to read back the data and correct certain errors.
And this is in relation to, up until now, the data on DNA has been used to store unstructured files versus the tool that you've developed. You've been able to integrate data storage in relational databases, and you've also used it to archive a 12 KB TPCH database to DNA as well.
Indeed, right. What we could have done is the standard approach. You just take the table, the relational table, and you say, well, that's just like a file. Yeah. We just put it on these oligos, add information about how to reconstruct the file in the end, and then read it back. That's kind of the classical way. But we thought, we can do this more clever, and essentially it's kind of exploit the structure of the data to keep it as small as possible, right? That's how we adapted kind of the tools, the existing tools already. But they fit very well into what the database tools are already.
And so just going back to that, how is it relational with the things like SQL joins and oligo? And you've tapped into this area of biochemical computing as well. Does it all come together in this step then?
Yes, indeed, right. So, it comes together in the sense that we're trying to emulate or replicate the functionality of a database system, right? We have then thought, with the database join, for example, that tries to find records that have the same primary keys, right? And kind of glue them together in an abstract sense. And we tried to do this, or we did the same thing in DNA, right? And so we kind of wanted to make sure that when we put the data or the table, the relational table into DNA, that we can essentially have also the ability to then query the database. We do this using PCR and also have these join operations, right?
Okay. Can you just explain a tad bit more on the PG oligo dump and the PG oligo restore tools? Like, how are you running the PG oligo dump and the PG oligo restore? How is it being used in archiving and recovering tools, like the whole protocol in the process?
Right. So the idea of these two tools, and they exist as such in Postgres, right? They are used to take relational tables that are in the database management system in Postgres to get them out, archive them. That's the PG dump. And then after a couple of years, when we need the data again, when we want to query it, we basically use PG restore to take these archived files and put them back into the database management system, right? Okay. And what we have done is really to adapt these such that we can store the resulting data. I mean, when we extract it from the database, and we can store that in DNA. And then once we've stored it in DNA, we read back through sequencing and then restore it back into a table. And we've done this successfully, like you mentioned, with TPCH. a small subset of that benchmark.
Yep. And, performing operations on DNA, they require encoding, specialized encoding techniques, and that can generate oligos suitable. You've mentioned that for biochemical manipulation. But what does that essentially mean?
Well, when we say we want to perform certain operations on DNA, right? What we do is we exploit biochemical processes that already exist, like PCR, like annealing for the join operation. And unfortunately, we can't change these processes. These are processes that have been, nature has been developed over millions of years. So we're not going to be able to change them. So what we need to do is kind of try to adapt our encoding such that the operations work on our oligos, right? So for example, in a very simple example for the join, all we needed to do is to move the join key. We have to store that or somewhere on the oligo. And to make it work with our approach, with the biochemical process we exploit, we just had to put it at the end of the oligo, right? So that's we have to be a bit clever about how we position things on the oligo to make sure that these processes still work.
Okay. And does that kind of improve the error rate within storing the data then?
So not necessarily for the operations. I mean, the encoding itself is it's very important how we encode information and store it on the oligo. Certain positions on the oligo are more prone to errors than others.
Okay.
So we need to be a bit clever about that. But then also when it comes to errors, what we mostly do is we develop new error correction techniques to compensate for errors, right? Rare errors are not that frequent, in my opinion anyway, when it comes to synthesis, sequencing, and storage, but they exist and we need to compensate for that.
But in all the other solutions that have been developed to the day, they involve heavily replicating the data and having loads of backup to, prevent the event in case of, there's an event of data corruption, just to kind of avoid that. But your technique relies on metadata. So is that cutting down the error, or are there just like the protocols that you're developing within OligoArchive, all in all, they go hand in hand, that, you're kind of going from step one, making sure there are no errors in the first place?
So, we have to tackle this on every level, right? We need to make sure, of course, that the underlying technology, synthesis and sequencing, have as few errors as possible. Still, errors will happen. We have developed error correction codes as well to simply make sure that if there are errors, we can compensate for that. But also with kind of exploiting the structure of the data helps us to reduce the need for error correction as well. Because like I mentioned this previously, this data cleaning technique, for example, that are well known in databases, have similarities to what we need to do for sequencing or for data that's coming from sequencing. So we can exploit that also, that we can reduce the errors there.
And how exactly are you doing it? Is it by adding bit of data into the blocks, or what does that process involve?
It's a bit of both, right? Yeah. So we add information, right, the error correction codes to protect against errors. But then also when reading out, we need to, as you mentioned, right, the information is available a couple of times. So we have the same information stored in multiple molecules. And in the data cleaning process, we can exploit that to align that and to reduce the number of errors.
Okay. Well, I guess, like they say, a bit of metadata can go a long way. But, in a lot of papers, headlines, wherever you go, there's a lot of talk about the density of DNA and the fact that it's capable of storing 215 petabytes in a single gram of DNA. But you've spoken about this in the papers as well. The Crider's rate for DNA is theoretically 0. And this is kind of fixed. It's biologically fixed. But how does that kind of stand against all of the other existing medium? And if you just want to explain what Crider's law is, just to kind of give more context.
Oh, it's basically how small we can store information or how compact we can store information or data. And it's typically measured against disk and data stored in disk. And the idea that every couple of years we can store the data in less space, right? That's basically the idea behind cry this rate. And DNA is massively more compact. I mean, the potential is considerable. It's about 10 million times smaller, 10 million times smaller than existing tape drives, right? Or even more. I think it's about 100 million times smaller than disk drives. So it's extremely compact.
And what does that mean then on, using it as an archival storage medium? What does that mean for, archiving data on DNA? Does
Oh,
that
it means that we can basically store the same amount of data in much smaller space. That has implications on also data centers, right? Today we store or archive our data in data centers, which are massive buildings. And if we can shrink this, we're not going to be able to shrink this by a factor of 10 million, but we can still, the smaller these data centers are, obviously the better, the less energy we need to run them in general. So yeah, that's pretty much the benefit there, right?
Do you think DNA data storage is going to run parallelly with the existing archival methods, or do you think by overcoming the challenges, we'll dive into that as well, we'd be able to develop solutions that are parallelly working, that you do have data centers, but at the same time, the cost or whatever factors that are hindering the progress in this field may be able to overtake and be like, all right, that's our archival storage medium?
I'm thinking that at the moment, the first step will be to introduce a new kind of storage medium. DNA is a new storage media that complements the others for the time being. It's really, given the access speed, the cost, this is really for data at the moment or in the foreseeable future, for data that needs to be stored decades. If it has to be read in tomorrow again, then it's not worth putting it in DNA. So I really think that will be the first use case for DNA. Later on, as technology improves, maybe the access speed will improve, the cost will lower, then it might move, maybe it's going to replace optical storage, maybe tape, maybe disks at some point. But for the time being, I think for the first step is really going to be an additional form of storage media for long-term, very long-term archive.
Which is complementing the current storage systems as well then.
Indeed, at the moment, I see it as complementary, right?
Yeah.
Today, for example, if we want to store something for, say, a couple of decades, we put it on tape, and then every couple of years we need to move it between tapes, right? We need to copy it over, etc., which is a costly process and so on. And so there, I think that's where DNA will fit. That we basically then write it once on DNA, and in 50 years we'll read it back and so on. So it will eat a little bit into the tape market, but it won't replace it anytime soon.
Which probably also means it's going to be environmentally sustainable as well, as opposed to replacing the tapes, which probably, are being thrown away, or like being replaced. It's just going to be, it's maybe a while of DNA with all the information with a couple of copies around the entire office. Like everyone gets to have their own archival. Here's your archive data, keep it on your table, and it's going to be safe, or however the companies go about it. But talking about the storage speed, has it been getting better even within the project, the OligoArchive project? Have you seen the progress happening from day one to now?
I mean, it's getting there, really. So when we look at sequencing, for example, I mean, reading back the information has become much faster and much cheaper, even in the last couple of years, right? During the runtime of the project, right? So there's clearly progress. And I feel like sequencing, in terms of technology, is almost where it needs to be. So in the next couple of years, it probably will get where it needs to be in terms of access speed and in terms of cost. When we look at synthesis, that's where progress has been made, and not least thanks to Helix Works, of course, in terms of speed and in terms of cost, right? But there, I think it's going to take a little longer to get it where it needs to be.
And in terms of improvements, what areas do you think let's just talk about the end-to-end product. what areas do you think we'd be seeing? That you just mentioned DNA synthesis has been an area of improvement, but what areas do you think we'll see or would need a bit of an improvement, and what does that timeline look like?
So I'm thinking that really synthesis is crucial, right? And because when we look at the workloads or the data that we want to store in DNA or for the long term, they're typically characterized by the fact that the data needs to be written, right? But it may not be read ever. It might be, we need to have it around for reasons of compliance or similar, but it's unlikely to be read. So in that sense, the write needs to be fast and cheap, and the read is not that important, right?
Yeah.
So synthesis is crucial, and we need to see more improvements there.
Yep.
Yeah.
And again, in terms of compliance, I definitely agree. With different industries, they have different compliances. With the healthcare industry, it could be a certain number of years that they need to keep the data within their data management system. But what does the timeline look like where you see that DNA data storage might get integrated?
I change my opinion every day. So today, I'll say it's between 3 and 5 years for the first commercially viable prototypes, I believe.
Yeah.
I think that's realistic. Like I mentioned, right, sequencing in terms of speed and cost, I think it's almost there. Synthesis needs to come along. There's really creative approaches there that kind of depart a little bit from how synthesis works for the life sciences. So I think 3 to 5 years is realistic for first prototypes, I think.
Yeah. And what are some of the challenges do you think that are actually hindering the progress? One, of course, like the DNA synthesis, the sequencing bit, but what do you think are the challenges you immediately need to come over so you can actually take the foundation to the next level? Because I feel everyone who's working on DNA data storage right now, everyone's building the base, like this is going to be the basic protocol that we build on. What would be some of the factors that are hindering the progress right now?
That's an interesting question. Yeah, what are the factors hindering progress? I think it's basically at the moment, like you said, we're building the foundations. Everybody's trying a little bit, and it's shown to work, right? We're there in terms of going end to end, at least in OligoArchive, we've shown that this is possible. What we need to do now is scale it up, make it a viable alternative. And it's really, I think we need to get the smartest people together and work towards that goal. I don't see any fundamental challenges.
Yeah.
The science says, when we've shown it works, we just need to scale it up in a meaningful way. What makes it a little bit harder, and it's going a little bit back to the synthesis, the point about synthesis being somewhat the roadblock at the moment, is that synthesis has been developed for the life science applications, and there's simply not enough demand in the life sciences for synthesis to kind of push this, to and make it cheap enough for our purposes. At the same time, we're also seeing another interesting effect, and when it comes to this, in the sense that for the life sciences, synthesis needs to be 100% accurate.
Yep.
Data storage, not necessarily. As long as we know what the level of errors is, we can compensate for that. So we are also a little bit at a point where we need to see if and how we can depart from DNA synthesis for life sciences and DNA synthesis for data storage.
And just out of curiosity, you mentioned, and now there are like loads of projects that are kind of working and everyone's kind of in their own sort of space. Let's just talk about encoding and decoding tools and technologies. How is that different from each sort of research group? Is everyone kind of working on a different protocol right now and just trying to figure out like, this is probably the most efficient ways? Everyone of course, the end goal is the same, but everyone's probably taking another route or with some overlaps.
Oh, absolutely, with some overlaps. But yes, everybody has a slightly different angle, right? So, for example, in OligoArchive went down the route and said, there's a different requirements for imaging data where we also can tolerate a bit of error versus other types of data like databases where everything needs to be precise, right? And then that means we have slightly different encodings, right? But overall, yeah, you're absolutely right. I mean, there's a lot of groups working on it. Everybody has a slightly different angle. Everybody's coming with a slightly different encoding. And what we need to be a little bit careful as a community is not to replicate work too much, right? And also, I do feel that everybody is looking also at, I'm working with this specific sequencing technology. That's why I come up with a new encoding. And that's perfectly fine. But at some point we kind of need to bring everything together, all the work together and come up with a unified approach in a sense.
Yeah. Just on that again, in terms of the sequencing part of things now. So, are there a lot of sequencing mechanisms out there, or like the DNA sequencers, and traditionally are they different from what you're using, or or is it more like the platform kind of remains same?
No, I mean, there has been it's quite interesting. No, there are a few different technologies out there, very few, a handful, maybe three, I would say, in principle. And each technology is driven by one company. Historically, there's been a lot of Illumina has been covered a lot of market share by sequencing by synthesis. And in recent years, Oxford Nanopore came out with a new approach, and that has been fantastic in the sense that that really made a difference in terms of price, accuracy, and speed of sequencing, right? So they basically developed this nanopore approach where you pull the strand, the oligos through a little nanopore and see how it disturbs the ion flow, and by that you can tell which nucleotide is passing through the pore. And so that's been a fundamental breakthrough, right? And that has really made a huge difference also for DNA storage, driving the price for sequencing down. Yeah.
And just talking about the costs, so what are some of the factors on which how can we make the whole DNA data storage, DNA as a medium for data storage, much more cost effective? What would be some of those areas where we definitely know? Again, I know DNA sequencing and DNA synthesis being quite the centric factors.
Yeah, absolutely. I mean, we basically have the synthesis, the storage, and the sequencing, and the storage we assume doesn't really cost a lot. So yeah, this is really the sequencing and the synthesis. We just need to make synthesis more cheaper. There's a couple of ideas out there really adapting the synthesis for data storage, introducing more errors, reusing reagents, these type of things that can be done. We just simply need to, we either go down the route of saying, oh, we're going to have so much more demand for synthesis through data storage that we can drive the price down, or we just work on the technology.
Absolutely. But if we can use DNA both as a storage medium and a computing resource, the implications can be beyond our imagination. Just on the research side, what impact could come from research labs that are working on DNA data storage?
I think, the research labs at the moment, particularly in academia, I think they're just ideally positioned to drive this forward because we can take risk. We can have negative results. These are still results which companies cannot easily afford to do. So we can really drive it forward in terms of fundamental research, trying out new and crazy ideas. That's our role, and that's where I think we can have the most impact. Basically what we want to do or can do is to de-risk the technology for companies to get involved as well.
Yeah. And that's quite interesting because that would be the approach traditional organizations would be allocating to startups, that startups are much more well equipped to take that risk and go out and develop something that traditional organizations cannot do because they would have to go through all the management processes and get the approval. But I like how the collaboration now within the consortium, and it's really nice that the European Parliament is coming forward and putting out grants and efforts in this area to put forward new and disruptive technologies that, the research labs, the spin-outs, everyone's able to come together and work on something really disruptive, and like the risk gets equally distributed. It's not just on one entity. So when you are looking to hire PhDs or postdocs to work on such projects or just even for DNA data storage, what is it that your lab looks for? Because this is it's not even a decade to this field. What is it that they should be skilled with?
It is a challenge to hire in that field. surprisingly so. It's rare to find students which have the right mix of kind of computing background and also a bit of biology background, right? So that's very difficult to find. I do find though, as time progresses and the technology itself, it's not as crazy as it used to be a few years ago, where people and PhD students and postdocs felt like, why would I get into this? It's a high risk research direction. Now it seems a little bit to be less high risk, so people are more interested in it. But like I said, it's I find it challenging to find people who have the exact right background. But it also, in my experience, doesn't matter that much. The most important thing is simply that the people who are interested in this and work on this, the people we want to hire, they're basically curious, driven and curious, and not kind of have a willingness to learn and are not scared of getting their hands dirty. And when I say this, I mean it's literally dirty. In the lab, in the end, yes.
No, that's true. I'd actually like to know a bit more about your experience when when you started talking about DNA data storage. What was it that people perceive? Because every time I speak about DNA data storage, people are like, oh, some medical data, big data, like genomics. And I'm like, no, this is using synthetic DNA to actually store data. Like you can put your photo, you can put a music file. So did you have those kind of resumes coming in talking all about the big data and genomics of kind of worked, and you were like, well, it's really not that, but this is what we're looking at?
Indeed, right? And also, that used to be when we started work on that a couple of years back, that used to be very common. And that's why I started to talk about DNA data storage rather than DNA storage, right? And that helped a little bit. Now it's easier to talk about it, but back then it was, I remember also when we were talking about pushing computation into DNA, kind of working using biochemical process to do computation on top of it. The reaction was like, I mean, are you crazy? Why would anybody do that? And so it's become more and more accepted. So that's good to see as well, right?
No, absolutely. So who should reach out to Scale Lab, and what would be the best way to reach out to Scale Lab?
Oh, whoever is interested in the topic, by all means, get in touch. The best way is just send an email, a message. We have it's on the website and elsewhere. Just pop a message generally, I get sometimes people interested to join the lab. Sometimes I get people who want to know more about the technology, how that would fit for them, or for industry, for industrial applications, and so on. We're open to discuss anything really.
Perfect. We will also put the link in the description, but this is just the beginning of a journey, and we've seen some massive breakthrough in just a decade, and plenty of things are still left to discover. Thank you so much, Thomas, for joining us today and sharing some of your research insights. It was great to have you on the show. We wish you and the rest of the members involved in the OligoArchive project every success in the project, and we can't wait to see what amazing results come out of it.
Thank you very much. Thanks for the opportunity.
Thank you for listening to this episode of Dive into Deeptech MoSS, powered by HelixWorks Technologies and brought to you from Republic of Work Studio, Cork's leading innovation hub. Follow us for more updates. Until then, stay tuned.